Here is the SRA database, Sequence Read Archive data. So these are all the little tiny bits of usually big genome or transcriptome sequences all in a pile. It's very nice. So you can take your SRA, once again, is just one of the choices on here. So we could go back to nucleotide right there. We go to protein. We got SRA. Yeah, it's nice. They're all on one spot and they're all connected. So the SRA databank's technically outside of GenBank. That's a separate thing that actually has those genes more or less on it and genomes. But yeah, there's useful stuff here. Now, as for accessing it, you can access it this way. So let's type in the name of a bug. And do a search, and there we go. And so this is for a Henocepilacna bug. And you can get, boom, click on any one of these. Inside of it will be several things. One is a thing called a sequence read experiment, SRX. Those are going to be, an experiment can have one or more runs in it. So they're just connected intellectually together. An experiment can actually have multiple species in it. You can say, as an experiment, I'm going to sequence everything in my backyard. And that's okay. It would all be in one SRX, and you'd sort it out later. But it'll have useful metadata here, which is going to be what sort of machine was used to make it. What does the sequence data look like? So this is paired end sequence data. And you can actually go to the run itself. And there's the run. And if you go to the run, it'll take you to a separate page. And there it is. You can download that 2.3 gigabyte data set, which contains 8 billion, I guess, nucleotides in 26.7 million pieces. So that's what the spots are. And yeah, anybody can download it, or you can blast it or anything like that. And it'll have some of that metadata down here. Every one of those is going to be associated with a bio project, and there are various other ways that the data gets grouped together. Yeah. And so it says this run has 2 reads per spot. That's what paired end reading means. And so the length is going to be 150 for each one of these. So it's little tiny bits of 150 nucleotide sequence right in there. And there are 26.7 million of them. We're not going to look through these one by one because that would be insane, but we have ways to search them all at the same time, and those are powerful. All right, there you go.